We're going to look at oxygen transport in the blood. Oxygen is transported in the blood in two ways. This is really important to know, so make sure you star this in your notes. A small amount of the oxygen is dissolved in the plasma. This is only about 1.5%. And that's because oxygen is not very soluble in the blood. It really does not want to be dissolved there. So we had to come up with a different way to transport the majority of oxygen in the blood. So most of the oxygen will be bound to hemoglobin in the red blood cells. So it's about 98.5% of the oxygen. And remember, oxygen binds to the iron in the heme group reversibly. So when the hemoglobin is not carrying oxygen, it's called deoxyhemoglobin. And when oxygen is loaded onto the hemoglobin and it's bound to the heme groups, then it's called oxyhemoglobin. And if we take a look at this chemical reaction, which way does the reaction go in the lungs? So if you remember the law of mass action, so if you have more of one thing on one side, it's going to push the reaction to the other side. So in the lungs, we have lots of oxygen in the inhaled air. So that's going to favor the reaction going to the right and loading oxygen onto the hemoglobin, which is what we want to do in the lungs. In the tissues, though, the oxygen levels are very low. So the oxygenated blood coming into that tissue is going to unload the oxygen at the tissue, which is what we want. And then answering this question at the bottom, how many oxygen molecules can one hemoglobin molecule carry? So remember that there are 4 heme groups on each hemoglobin molecule. So one hemoglobin molecule can carry 4 oxygen molecules. And when the hemoglobin is full of oxygen, there are 4 oxygen molecules bound to it, then we say the hemoglobin is 100% saturated. These pictures just show the loading and unloading of oxygen. So remember, in the lungs, the partial pressure of oxygen in the alveolar air is much higher than in the capillary blood. So oxygen will diffuse across that respiratory membrane, and most of it will bind to the hemoglobin that's in the red blood cells. And then at the tissues, where the oxygen levels are much lower than what's in the blood coming in, oxygen will just diffuse off of the hemoglobin and into the tissue. So we'll unload the oxygen at the tissues. There are several factors that affect hemoglobin's affinity for oxygen. So affinity is just meaning how well the hemoglobin binds to the oxygen, and we call this hemoglobin saturation. Let's look at the effect of the partial pressure of oxygen on hemoglobin saturation. The higher the partial pressure of oxygen, the more oxygen is going to bind to the hemoglobin. When one oxygen binds to the hemoglobin, it makes it so the hemoglobin is more likely to bind even more oxygen molecules. If we take a look at the pulmonary veins and systemic arteries, where the partial pressure of oxygen is 100 millimeters of mercury, the hemoglobin is going to be 98.5% saturated. And then the lower the partial pressure of oxygen, the more oxygen will dissociate or fall off the hemoglobin. So at a partial pressure of 40 millimeters of mercury, which we see in the systemic veins, after it's just gone through the tissues, the hemoglobin is going to be 75% saturated. This picture shows a red blood cell, and the squares inside are hemoglobin molecules, and the blue dots are oxygen molecules. So this is in systemic arterial blood, and you can see that most of the hemoglobin molecules are carrying 4 oxygen molecules. There's a few that aren't. So the hemoglobin is 98.5% saturated in systemic arterial blood. This red blood cell has already done gas exchange with the tissues and is now in the systemic venous blood. And notice that the hemoglobin is 75% saturated. So we've dropped off about 25% of the oxygen that the red blood cell was carrying at the tissues. So there's still quite a bit of oxygen that is still bound to the hemoglobin. So there's a large reserve of oxygen that if you start exercising or tissue demand increases, you still have a lot more oxygen that can be dropped off at the tissues. This is the hemoglobin-oxygen dissociation curve. This is a really important graph to know, so make sure you star this. On the bottom, we have the partial pressure of oxygen, and on the y-axis, we have the percent oxygen saturation of hemoglobin. In the lungs, the partial pressure of oxygen is high at 100, and in the lungs, we're going to load oxygen onto the hemoglobin until it's 98.5% saturated. In the tissues, the partial pressure of oxygen is 40 millimeters of mercury, so it's much lower. So in the tissues, we're going to unload the oxygen until the hemoglobin is about 75% saturated. The normal hemoglobin-oxygen dissociation curve in the middle, that's dark blue, can be shifted either to the right or to the left. So this is really important to know, so make sure you star this. If you shift the curve to the right, that's going to decrease hemoglobin's affinity for oxygen. It's not going to bind to it as well. You're going to unload more oxygen in the tissues, and you're not going to be able to load as much oxygen in the lungs. So if we look at the tissues where the partial pressure of oxygen is 40, normally if the curve is not shifted, the hemoglobin saturation would be 75% after going through the tissues. But if we shift the curve to the right, we end up dropping off a lot more oxygen at the tissues. So maybe we're at 45% oxygen saturation of the hemoglobin. And then in the lungs, we're not going to load as much oxygen on. So at a partial pressure of 100, normally we would be 98.5% saturated. But with the right shift and not binding to oxygen as well, maybe we're going to be at maybe 93% oxygen saturation. If the curve shifts to the left, we're going to increase hemoglobin's affinity for oxygen. So it's going to bind to the oxygen much better. So you're going to be able to load more oxygen in the lungs, but you're not going to be able to unload as much oxygen in the tissues. So if we look in the lungs at a partial pressure of 100, if the curve is shifted to the left, the lighter blue color here, you can see that our oxygen saturation will be much higher. Maybe it's at 99.5% instead of 98.5%. But if you shift the curve to the left in the tissues, down here at the 40 millimeters of mercury, you can see that you're not going to drop off as much oxygen. So maybe your oxygen saturation after going through the tissues is 80%. So the reason these shifts in the curve are important is because we want to, in the tissues, shift to the right, because that means we're going to unload more oxygen. And then the lungs, we want to shift to the left, because that means we can load more oxygen in the lungs. There are a number of things that will shift the curve to the right or to the left, and temperature is one of them. At the normal body temperature of 37 degrees Celsius, here's the normal hemoglobin-oxygen dissociation curve in the middle in dark blue. But if you start exercising, those active tissues will increase the local temperature around them, and that will shift the curve to the right, which is actually good because in the tissues you do want to shift to the right, because then you can unload more oxygen at the tissues because hemoglobin isn't going to bind to it as well. So at the tissues of a partial pressure of oxygen of 40 millimeters of mercury, you can see that if you shift to the right, the oxygen saturation ends up being around 45%. So you're going to end up dropping off a lot more oxygen than if you were just at the normal body temperature and hadn't shifted the curve to the right. So normal body temperature, 75% oxygen saturation of the hemoglobin. In the lungs, generally the air that we're breathing in is going to be lower temperature than the body temperature. So the lower temperature will shift the curve to the left, which is good in the lungs because that means we're going to increase the affinity of hemoglobin for oxygen. It's going to bind to it a lot better and increase the loading of the oxygen in the lungs. So in the lungs, the partial pressure of oxygen is 100. And you can see that by shifting the curve to the left in the kind of gold color, you can see that we're going to get a higher oxygen saturation than if we hadn't shifted it to the left. So shifting the curve to the left with the lower temperature in the lungs will increase oxygen loading of the hemoglobin in the lungs. pH will also shift the curve to the right or to the left. This is called the Bohr effect, just named after the scientist that discovered it. The way I remember this is that the word Bohr has an H in it, and so does pH, so those two go together if that helps you. So pH will affect hemoglobin's affinity for oxygen, or how well it binds to the oxygen. So if you look at this chemical reaction in the middle, when oxygen binds to the hemoglobin, it will kick off one of the hydrogen ions from one of the amino acids in the protein part of the hemoglobin. And then the reverse reaction can also occur where the hydrogen ion will bind back to the protein part of the hemoglobin and help kick the oxygen off and unload it. So when tissues are active, they're going to be generating a lot of carbon dioxide because they're breaking down lots of glucose to make lots of ATP. And this chemical reaction on the bottom left, we're going to be looking at a lot more later. But if you increase the amount of carbon dioxide, that will push the reaction to the right, and notice that you generate lots of hydrogen ions. And if you increase the hydrogen ions, that decreases the pH and makes it more acidic. So when tissues are active and exercising, you're going to be generating lots of carbon dioxide, which will generate a lot of hydrogen ions. And that will decrease the pH. And if the pH decreases and becomes more acidic, that will shift the curve to the right. And with all of these hydrogen ions running around in this active tissue, the hydrogen ions will then bind to the protein part of the hemoglobin and help kick the oxygen off to unload it at the tissues. So in active tissues, when the pH decreases, becomes more acidic, you're going to be unloading a lot more oxygen at the tissues. So now your oxygen saturation is about 45%. Whereas if you had not shifted to the right, then the oxygen saturation would be around 75%. In the lungs, there's not a lot of carbon dioxide because the air you're breathing in doesn't have a lot of carbon dioxide in it, and you're exhaling that CO2. So if there's not a lot of carbon dioxide, that's going to turn this reaction and shift it to the reverse, to the left. And if you do that, notice that the hydrogen ions will disappear by the time you get all the way over here on the left. So that will actually increase the pH or make it more basic. So in the lungs, we're going to shift to the right, and that will increase hemoglobin's affinity for oxygen, and we're going to bind more oxygen to the hemoglobin in the lungs. So that's good because that's what we want to do. So that's the Bohr effect, just pH affects hemoglobin's affinity for oxygen and will shift the curve either to the right or to the left. So it shifts to the right in the tissues, shifts to the left in the lungs. The partial pressure of carbon dioxide will also affect how well hemoglobin binds to the oxygen and will shift the curve to the right or the left. Carbon dioxide will bind to the amino acids on the protein part of the hemoglobin, and that will form carbaminohemoglobin. The reason it got that name is that the carb refers to carbon dioxide, amino refers to the fact that the carbon dioxide binds to the amino acids in the hemoglobin. When carbon dioxide is bound to the hemoglobin, hemoglobin is not going to bind to oxygen as well, and this is called the carbamino effect. So in the tissues, you have a lot more carbon dioxide because you've been generating that from making ATP, and more carbon dioxide will bind to the hemoglobin in the tissues, and that means that the oxygen will not be able to bind as well, and so will increase the oxygen unloading, especially if the tissue is more active because you're going to have more carbon dioxide being generated. So that means that if the oxygen is not binding to the hemoglobin as well, we've shifted the curve to the right. So if you look at their curve here, if we shift it to the right, the purple, you can see that we're going to unload more oxygen at the tissues. Maybe we have 45% oxygen saturation. Then if we hadn't shifted the curve, we would have been at 75% saturation. In the lungs, just the opposite happens. So in the lungs, there's less carbon dioxide, and if less carbon dioxide is bound to the hemoglobin, more oxygen can bind to the hemoglobin. In the lungs, we're going to shift the curve to the left. So if the partial pressure of oxygen in the lungs is at 100, you can see that shifting the curve to the left, in the lighter blue here, we're going to be able to load more oxygen on than if we hadn't shifted the curve. So that's another example of the Carbamino effect. The chemical BPG also will affect how well hemoglobin binds to the oxygen, and this will shift the curve to the right or to the left. BPG stands for 2,3-bisphosphoglycerate, which I will not ask you about, just know BPG. And BPG is produced during glycolysis as the red blood cells do fermentation. So remember, that's how they make their ATP since they don't have any mitochondria. So if we take a look at the chemical reaction on the bottom left here, you can see that as the glucose is being broken down to make the pyruvate and finally the lactic acid, BPG is just one of the intermediates. And if BPG is being made and there's a lot of it around, it will shift the curve to the right. So it will not allow the hemoglobin to bind to the oxygen as well, and so we're going to increase the unloading of oxygen at the tissues. So in the tissues, we're going to be producing lots of BPG, and that will shift the curve to the right, and that will increase the oxygen unloading at the tissue. But in the lungs, we're going to shift to the left, and that's because in the lungs you've got lots of oxygen binding to the hemoglobin, and the oxyhemoglobin will inhibit this enzyme that's used to make the BPG. So in the lungs, the BPG is not going to be made, and so that will shift the curve to the left. And that's good because in the lungs that will allow you to load more oxygen onto the hemoglobin because the hemoglobin is binding to the oxygen better. The last thing that affects how well hemoglobin binds to oxygen is carbon monoxide. Carbon monoxide is found in things like exhaust fumes, and it will compete with oxygen to bind to the heme group in hemoglobin. So if you're breathing in a lot of carbon monoxide from exhaust fumes, the carbon monoxide will outcompete the oxygen, and not very much oxygen will then be able to bind to the hemoglobin because it's all full of carbon monoxide. And that means you're not able to deliver enough oxygen to your tissues, and now your tissues can't make enough ATP, and then that will kill you. Symptoms of carbon monoxide poisoning include usually you get a headache first, and then you might feel dizzy, nauseous, tired, and then you become unconscious, and if you aren't gotten out of there, then that will kill you fairly shortly. Carbon monoxide poisoning patients that come into the hospital, how we treat them is they will be given 100% oxygen through like a nasal cannula. If that's not enough, they can actually do hyperbaric oxygen chambers, because the higher pressure of the oxygen in those chambers will drive more oxygen into the blood, and that will outcompete the carbon monoxide. And then lastly, they can also do a blood transfusion, because if you get fresh red blood cells in there that are not bound with carbon monoxide, they can then carry the oxygen to your tissues. And then just a summary of the factors that affect how well hemoglobin binds to the oxygen. Make sure you star this and know this, because this would be a great multiple choice question on the test. In the lungs, remember that you want to shift the curve to the left. And if you shift the curve to the left, hemoglobin is going to bind to the oxygen better, and you can increase oxygen loading. So factors that are occurring in the lungs to make the curve shift to the left: you have high partial pressure of oxygen, pH is higher, it's more basic, you have less hydrogen ions. The temperature is cooler because of the air that you're breathing in is not as warm as body temperature usually. Partial pressure of carbon dioxide is lower in the lungs, and also you're not making that BPG. In active tissues, like when you're exercising, we want to shift the curve to the right. And that means hemoglobin won't bind to the oxygen as well, and we're going to increase oxygen unloading in the tissue. And that will happen when the oxygen levels are lower in the tissues, the pH is lower or more acidic, you have more hydrogen ions. temperature is increased from the exercising. Partial pressure of carbon dioxide is higher, and also BPG is higher. So all of those things will shift the curve to the right.